Okay, let's unpack this. Welcome to the Deep Dive, your shortcut to being truly well-informed. Today, we're plunging into a critical new finding. It could fundamentally reshape how we approach cancer treatment. We're talking specifically about non-small cell lung cancer, or NSCLC. Now, immunotherapies, they've brought incredible breakthroughs, really revolutionizing cancer care, no doubt about it. But here's the persistent challenge, one that deeply affects patients and clinicians alike. How do we know which ones will truly benefit from adding specific, powerful treatments to their existing immunotherapy regimen? It's a huge question, often frustrating.
Exactly, it really is. And our mission today is to explore a pre-planned analysis. It comes from a significant phase 3 clinical trial, famously known as the APLE study. And our source for this deep dive is a very recent scientific article published in Nature Communications. It focuses on a potential new biomarker that could finally provide an answer, or at least part of one.
Right. So our core objective really is to give you a clear understanding. How might we identify those NSCLC patients, the ones who will genuinely benefit from adding a particular therapy? We're talking about bevacizumab here, adding it to their chemoimmunotherapy regimen. It's about getting smarter, more precise. Okay, let's start by setting the scene a bit. The landscape of NSCLC treatment. Immune checkpoint inhibitors, ICIs, you hear about them a lot. Things targeting the PD-1 or PD-L1 pathway. They've had an enormous impact. I mean, they're now established standard treatment, not just for metastatic disease, but even earlier in the perioperative setting. They've completely changed things.
Totally. And that's where bevacizumab steps in. It's an antibody, and it targets something called vascular endothelial growth factor A, or VEGF-A. Now, bevacizumab has been a promising candidate for combination therapies for a while. And in fact, it's already used with ICIs as a standard option for several other cancer types, including NSCLC, actually, and hepatocellular carcinoma. But what's been particularly challenging, though, is this. Despite its existing use, there hasn't been a clear biomarker, nothing to reliably identify patients who are likely to experience a significant survival benefit when you add bevacizumab to their immunotherapy. So this raises a really important question, doesn't it? How do we select the right patients instead of, treating everyone and just hoping for the best?
Okay, so this is where it gets really compelling, I think. VEGF-A. What is it about VEGF-A that makes it such a crucial player in this whole puzzle? What exactly is it doing?
Yeah, VEGF-A is a fascinating molecule. It's a cytokine cheery. You can think of it as like a signaling molecule. It's produced by both the tumor cells themselves and even our own immune cells. Its role is complex, but critically, it plays a major part in creating an immunosuppressive tumor microenvironment.
Immunosuppressive, meaning it dampens the immune response.
Exactly. Imagine it like a master conductor, but for putting the brakes on the immune response. It essentially hinders our immune system, makes it harder for our natural defenses like T cells and other key immune players to get activated, harder for them to identify cancer cells, and harder to effectively kill them. It also kind of helps create a protective shield around the tumor, makes the environment more favorable for cancer growth innovation. So the theory is pretty clear. If you inhibit VEGF-A, you should, in theory, take those brakes off. And by doing that, you increase the efficacy of the immunotherapy by counteracting these immunosuppressive effects.
Okay, so if I'm getting this right, VEGF-A is like hitting the brakes on the immune system near the tumor. And blocking it with something like bevacizumab is trying to release those brakes.
That's a good way to put it, yeah.
That sounds like a powerful mechanism.
Yeah.
Okay, so with that understanding of VEGF-A, let's turn to the study at the heart of this deep dive, the APPLE trial. This was, as you said, a randomized phase 3 trial, designed specifically to evaluate adding bevacizumab to standard treatment, which was platinum doublet chemotherapy plus 8-izolizumab, that's a PD-L1 inhibitor. And this was for patients with metastatic or recurrent non-squamous NSCLC.
Right. And here's where the story takes that initial surprising turn.
Yeah.
The overall APLE study did not meet its primary endpoint.
It didn't work overall.
Not for the general patient population, no. The regimen containing bevacizumab was not superior. Progression-free survival, PFS, which is what they were mainly looking at, was actually quite similar. It was a median of about 6.4 months in the standard arm versus 7.7 months in the bevacizumab arm. Not a significant difference. So on the surface, that was, well, a disappointing result.
Which is precisely why this specific analysis becomes so critical, right?
Sure.
This wasn't just like a random look afterwards. It was a pre-planned exploratory analysis within that bigger APPLE trial.
Yeah. Exactly. Pre-planned.
They were specifically searching for a predictive biomarker, zeroing in on these baseline serum VEGF-A levels and its specific isoforms, VEGF-121 and VEGF-165, using an ELISA system. Which is basically a simple blood test, right?
Essentially, yes.
So it's like a detective returning to the scene of a seemingly unsolved case, looking for that hidden clue.
A good analogy. And those clues came from analyzing samples from 149 patients from the original APLE trial. There were 76 in the chemotherapy atezolizumab arm, the control group, and 73 in the arm that also received bevacizumab. And importantly, this included patients with both EGFR wild-type and EGFR mutant tumors. That's a key distinction in NSCLC.
Okay, so after all that searching, after that deeper look, what was the breakthrough? What was the aha moment?
Well, the core finding was truly a breakthrough, I'd say. While the overall study showed no benefit for adding bevacizumab, its addition significantly improved progression-free survival, but only in patients with low baseline serum VEGF-A levels.
Low levels, that's the key.
That is the key. For patients with low total VEGF-A, adding bevacizumab nearly doubled their progression-free survival compared to the control group. We're talking about going from about 5.8 months up to over 10 months, 10.4 months specifically.
Well, it doubled.
Yeah, and this dramatic difference was statistically significant. It was also consistent across the different forms of VEGF-A they tested, including the isoforms VEGF121 and VEGF165. Similar results there. And this stands in stark contrast to patients with high VEGF-A levels. In that group, PFS didn't differ substantially between the arms. And actually, in some cases, it even tended to be shorter with bevacizumab.
Shorter. Okay, that's significant too. So a really powerful distinction. It highlights this crucial subgrouping. You mentioned EGFR status. This predictive power, it was particularly strong for the EGFR wild-type tumors.
That's right. It was particularly strong and confirmed for patients with EGFR wild-type tumors. So for them, having low levels of total VEGF-A or VEGF121 or VEGF165, it indicated a much more favorable PFS when bevacizumab was added to the chemoimmunotherapy. And their more advanced statistical analyses, the multivariable analyses, they strongly concluded that total VEGF-A, or TVEGF-A, should probably be the preferred biomarker, the one you'd measure preferentially among the three VEGFs for these EGFR wild-type NSCLC patients.
Okay, so TVEGF-A is the main one to look at for that group.
It seems so. But it's important to note, though, that no overall survival benefit was observed, which was actually consistent with the overall APLE study result. This particular biomarker analysis was focused specifically on progression-free survival, PFS.
Got it, PFS, not overall survival yet. Okay, so given that, how do these different isoforms like VEGF121 and VEGF165, how are they playing into this? Are they just slightly different versions telling the same story? Or do they give us something more precise? Can they refine patient selection even more?
That's a great question. They are indeed slightly different versions, arising from how the VEGF-A gene is spliced. And the study did look at whether one might be a better predictor or offer, more nuanced information. What they found was interesting. While most patients, about 70.5% overall, and even higher, 75.4%, in the EGFR wild-type group showed consistent levels, meaning they were either high across all three VEGFs or low across all three. There was a notable number, around 27.5% overall, maybe 22.8% of the EGFR wild-type patients who had discrepancies. High in one, low in another.
Okay, so some inconsistencies there.
Right. And what's potentially valuable is measuring VEGF165 in addition to the total VEGF-A. Here's why. In the high total VEGF-Y population, remember, that's the group where bevacizumab generally wasn't beneficial. If their VEGF165 level was also high, the tendency for even less benefit or possibly even worse outcomes from adding bevacizumab became even more evident.
So a double check almost. If both total and non-65 are high, it really looks like bevacizumab isn't the way to go.
It strengthens that signal, yes. For example, in the subgroup with both high total VEGF-A and high VEGF165, the median PFS was actually only 6.5 months with bevacizumab, compared to 9.7 months in the control arm for that specific subgroup. The hazard ratio there suggested potential harm.
Wow, so this isn't just about finding who will benefit. Crucially, it's also about identifying who won't, or even who might be harmed. That feels like a really practical takeaway. You could identify patients who should actively avoid adding bevacizumab, potentially sparing them from ineffective treatment, costs, and importantly, unnecessary side effects. That's a huge step for personalized medicine.
Right.
Okay, so ultimately, putting this all together, what does this mean for you, the listener, or someone maybe navigating these incredibly complex decisions in cancer treatment?
Well, I think one of the most important implications is the sheer ease of measurement. Serum VEGF-A assays, these blood tests, they are easy to measure, widely available, and so they have significant potential for, daily clinical practice. They could genuinely support treatment decisions, helping guide clinicians, pointing towards which advanced cancer patients, specifically those with low baseline VEGF-A, might actually benefit from this combination. It's also quite surprising, isn't it? Even counterintuitive, perhaps, that adding bevacizumab actually tended to shorten PFS in patients with high concentrations of VEGF-A.
Yeah, why would that happen?
Well, the researchers speculate a bit. It might be that those very high levels of VEGF-A could somehow prevent bevacizumab from effectively taking the brakes off the immune system. Or perhaps it makes the overall ICI treatment less effective because of bevacizumab-specific side effects. Things like hypertension, bleeding, hematologic toxicity. Maybe higher VEGF-A makes patients more prone to those toxicities, which then limits the effectiveness or duration of the immunotherapy part. So it suggests high VEGF-A might actively counteract the intended benefits or increase toxicity, making the whole approach less effective for those patients. And another crucial point revolves around that other group, the EGFR mutant subgroup. Remember, the original overall APELE study had hinted at a possible benefit for adding bevacizumab in these EGFR mutant patients. Their PFS looked maybe a bit better, but this specific biomarker study found no significant difference in baseline VEGF-A levels between the EGFR wild type and EGFR mutant patients. They look pretty similar. And crucially, low VEGF-A did not appear to be a predictive biomarker for bevacizumab benefit in the EGFR mutant patients.
So the VEGF-A level didn't predict benefit in the mutant group, even though there might be some benefit happening there.
Exactly. Which suggests that if there is a benefit for adding bevacizumab in EGFR mutant patients, it likely stems from a different mechanism, something VEGF-A independent, maybe involving other signaling molecules the paper mentions, like interleukin-8, transforming growth factor beta, CCL22. Other pathways might be more important in that context.
Fascinating. Okay, and as with any good scientific research, it's important we acknowledge the limitations, right?
Absolutely. Always important. The authors are clear about them. The sample size for this specific biomarker analysis wasn't statistically predetermined for this kind of subgroup analysis. It was exploratory. We also still don't fully understand all the genes that might influence someone's serum VEGF-A levels. And they didn't look at VEGF-A levels in the tumor tissue itself, only in the blood serum. Would that tell a different story? We don't know. And the biological significance of those discrepancies between the different VEGF isoforms in some patients isn't fully clear yet either.
Right, so definitely areas for more research.
For sure. These are all areas ripe for future investigation.
But what's truly fascinating here, I think, is how a therapy that, looking broadly, didn't show an overall benefit. It really shines when we potentially know precisely who to get it to. This deep dive reveals how a relatively simple blood test for VEGF-A could potentially be a game changer, helping to personalize treatment, optimize it for NSCLC patients, moving us away from that one-size-fits-all approach.
Absolutely. That's the promise here. The conclusion from the paper is clear. Low serum levels of total VEGF-A, or its individual isoforms, at baseline, they represent a potential predictive biomarker for selecting those NSCLC patients, specifically EGFR wild type, who are likely to benefit from this specific combination therapy. It's a very promising step forward, definitely. But, as we said, it really underscores the need for further confirmatory studies. We need to validate these findings in larger trials before this becomes routine clinical practice.
Of course, validation is key. But think about this. Just as a final thought for you, our listener, consider how many other seemingly failed clinical trials might hold hidden benefits, just waiting perhaps for the right biomarker to unlock their potential. What other broad medical interventions, treatments we use widely? What if they hold personalized keys to success? Keys we could find if we only knew where to look, and maybe what simple tests could guide us. Something to ponder. Thank you for joining us on this deep dive.